Okay, good morning, everybody. Can you hear me? I don't know if I'm on here.
You're ahead of me.
Am I on here? Okay. Well, thank you for being here. This is the Early Therapeutics and Rare Cancer Committee, and we have a very full agenda today. So we will get started and hopefully keep everybody on time. And so our focus is, of course, early therapeutics and rare cancers. And we've been especially focused on what I think is a huge unmet need, and that's the rare and ultra rare cancers. As you all know, by definition, rare cancers are uncommon individually, but altogether they make up almost 25% of the cancer burden. So it's a very large unmet need. What we're going to start with is Lara Davis. And Dr. Davis is associate professor at the Knight Cancer Center and co-leader of translational oncology. And she is leading our working group on sarcomas under the Early Therapeutics and Rare Cancer Committee, and will give us an update from that committee, from that working group. Thank you, Lara.
Hey, everybody. Hopefully we've got some virtual attendees as well. Great to see everybody. I'm Lara Davis. Thank you, Dr. Kurzrock, for inviting me to give updates on the sarcoma working group activities that have been going on lately. First, as a reminder, this is very much an open working group. We welcome anybody. I'll maybe caveat that with maybe not the industry folks for our working meeting that's coming up later today, but starting at 12:30 in Pacific F. You can also join virtually. Please come. It's going to be a working meeting. We want to hear new ideas. We're going to work through some old ideas. And then we also have our monthly meetings via WebEx that anybody is also welcome to join. I'm going to just throw out a little bit of a reminder that there are these positions called liaisons that each of the disease committees has a liaison to some of our other committees within SWOG. And I currently serve as the liaison for the AYA committee and the survivorship committee between this committee and that one. I'd like to just offer up, if there's any young investigators, either here in person or virtually, if somebody really wants to get more involved in SWOG and is really interested either in early therapeutics and rare cancers and survivorship, or AYA and rare cancers, please let me know, let Dr. Kurzrock know, and we may be able to have some opportunities for you that way. Essentially what the liaisons do is just go to both meetings and crosstalk. Make sure that we all know what's happening. So back to sarcoma specific information. So our working group formed in October of 2020, so about 2.5 years ago now, and we had these dual goals. Was to make sure that we're all collaborating and making sure to cross enroll on NCTN sarcoma concepts outside of SWOG, but then also to develop our own SWOG sarcoma concepts. And I do want to take a moment, because when we started, it became rapidly clear to me that nobody had any idea what was happening in the other groups. We didn't even know if there were committees for sarcoma in the other groups, or if they were working groups. We didn't know who they were. We didn't know why they were. We certainly didn't know what they were working on. So I figured out who they were and what they were, and put them to put us all together in communication. And believe it or not, that was really difficult. And now we have the chairs and vice chairs of all of our either working groups or committees across the NCTN meeting regularly, so quarterly, and then in person twice a year. I think that this hopefully is going to substantially increase our collaboration and communication. And we're actually expanding our in-person meetings to include our European and Canadian colleagues and the early therapeutics group and SARC. So there will be at least high level communication that hopefully will end up with improved collaboration. So there are 5 currently open NCTN sarcoma studies, none of which are SWOG led yet, but please make sure that you are aware of these trials within your institutions. Encourage cross enrollment. And so 3 of these open trials are from SWOG, but open to 40 or 50 years old. So the first just opened quite recently was a randomized phase 2/3 trial using our standard osteosarcoma backbone of MAP plus or minus cabozantinib. This is for either metastatic or non-metastatic at presentation first-line treatment for osteosarcoma. And champion Dr. Andy Livingston from MD Anderson is here in the audience. I think he's hiding behind a pole. And he'd be happy to tell you more about the status of that. We also have a randomized phase 3 trial of a surgical approach for osteosarcoma metastases. Dr. Hofstetter from the surgery committee is championing that. Again, that's a trial that is very important, but is struggling with accrual because many people feel strongly one way or the other of which surgical approach is correct. And given the fact that it's about 50-50, we know that we do not know the answer to that question, and that question needs to be answered in a prospective manner. So please encourage enrollment. And finally, for high-risk rhabdomyosarcoma patients open to age 50, and 2 alliance trials, both fairly recently opened. One is for metastatic uterine leiomyosarcoma, championed by Dr. Rusty Robinson, and very, very newly opened phase 2 for dedifferentiated liposarcoma. We'll hopefully identify a champion during our working group meeting today. And those are all open. There are others that are truly opening soon. I have actually had a couple of these on the opening soon slide for a few group meetings now. So representative of the extremely fast process. 2 from ECOG. One is the STRAS trial for high risk retroperitoneal sarcomas, either liposarcoma or leiomyosarcoma. And that has actually been open in Europe for over 3 years, which is why we thought it would be open here a lot faster. Soon, Dr. Maman and I are co-championing this. Do you have any updates?
Soon is all I've heard.
Exactly. It's soon. It's been at NCI for quite a while, I think, at this point. And then the ECOG Akron randomized phase 3 of surveillance, so chest X-ray versus chest CT, again, should be very soon, championed by Dr. Libby Davis. And the randomized phase 3 for metastatic Ewing sarcoma is very close, actually. Yeah, I'll be the champion of that because I'm on the committee. All right, so down to the meat, SWOG sarcoma concepts. We have been working hard on moving some concepts forward. First, I will remind everybody about Dr. Michael Wagner's angiosarcoma capsule that was actually disapproved about a year ago by CTAP. And the primary reason for that was slow down, guys. You need to wait for the metastatic randomized trial that Alliance is running to read out before we decide if the neoadjuvant approach is worth pursuing. I think as practicing angiosarcoma doctors, we're all pretty darn sure that it's extremely worth pursuing, but we are waiting for the data since that has fully accrued and at least parts of the trial are being resulted at ASCO. We should have that data quite soon. And then the additional kind of suggestions and contributions to the disapproval had to do with the complexity of the trial. So Dr. Wagner is working on kind of some ideas for how to streamline the approach and likely possibly revising it to be a neoadjuvant cutaneous only concept, something like that. And then working amongst the groups to come up with a way to make it the approach acceptable to most sarcoma doctors, which is always difficult when we have no standard of care. So our second major capsule that is being developed is by Dr. Jeremy Harris, who's joining us virtually today. This is a phase 2 trial, non-inferiority trial, extremely important question of whether or not short-course radiation is as good as the standard radiation that we have been using for decades. And this is for our high-risk soft tissue sarcomas. So these are high-grade sarcomas, not tiny sarcomas that are treated with curative intent, almost always treated with radiation, and lots and lots and lots of individual institutions have been conducting shorter course regimens for quite a long time, but we have never had a randomized trial, randomized prospective trial, and Dr. Harris has bravely taken this on. The hypothesis, of course, being that 5 fractions is not inferior to the standard 25 fractions. This schema has been basically the same since the beginning and has been debated many, many, many times. Dr. Harris has presented this to many investigated institutions and groups who all have their ideas of the best way to do it. Nonetheless, it comes back to this design that is both realistic and pretty much acceptable to pretty much everybody, which is working, essentially randomizing to either the short course of 5 fractions versus 25 fractions, stratifying these patients by tumor dimension as well as, greater than or less than 10 centimeters, as well as an upfront declaration of whether or not adjuvant chemotherapy will be given. So chemotherapy is not required. It is up to the treating investigator. The treating investigator could change their mind after resection, but if there's a plan to give chemotherapy, that would be a stratification. So this is a large study, 147 per arm of evaluable subjects. 117 evaluable subjects enrolling 147 per arm. And then post-treatment, if a RT boost is necessary or recommended, that is option, or as I mentioned, the adjuvant chemotherapy. Primary objective is evaluating local failure rate. This again has been debated back and forth. Should it be toxicity? But really what matters to the patient and the treating doctor is, does the cancer come back in that area? And so that is our final primary objective, with important secondary objectives and kind of general eligibility as described here. Historically, the 2-year local failure rate is about 11% with radiation. And so this non-inferiority design would allow up to an upper bound of 21% local failure. This is with those 237 evaluable patients. We anticipate it will be about 4 years to enroll based on prior the RTOG trials. And as mentioned, Dr. Harris has been working on this for a very long time. He has presented to multiple committees and groups, and outside of these formal presentations, he's also discussed with some of the larger groups their concerns and how he might get them on board. So it's been a valiant effort. And excitingly, we now have a number. So this is S2310. The executive committee approved mostly this concept, but with the significant concern of feasibility. So this is where I need to ask for your assistance. All of your institutions should have received a survey that says, would you enroll on this trial? Please say yes. And then we will be able to prove that everybody is invested and interested in this question. And then we'll be able to move forward with bringing this to CTEP. in addition, this is going to be a very hot topic of discussion amongst the NCTN, EORTC, everybody at the ASCO in meeting with all of the leadership. In prior meetings, we have discussed the possibility of a CDK4/6 inhibitor plus immune checkpoint inhibition for advanced dedifferentiated liposarcoma. It seemed like low-hanging fruit, a very obvious combination, but we were waiting for safety data readout. Apparently, that was not required because Alliance just opened this trial. So this has been abandoned and is no longer in development. Our other concept that we are working on is Dr. Cranmer's phase 2 of neoadjuvant abemaciclib in combination with radiation for retroperitoneal liposarcomas. This would be for patients with curative intent. And the current safety pilot run-in is almost-ish ready to start enrolling at single institution, at which point we will start the more formal capsule development here with the help of our fabulous statisticians, Megan and Anna. I'd like to highlight a really exciting opportunity. We were, as kind of a direct product of getting the NCTN group leaders together, sarcoma leaders together, we were approached by SWOG suggesting that we co-develop this pilot study. And I'm really thankful for the opportunity that Dr. Weiss and Dr. Mead from SWOG have seen that this could be a very mutually beneficial opportunity. And the hypothesis with this pilot is that the addition of MEK inhibition to our standard treatment for malignant peripheral nerve sheath tumor, MPNST, is that it would be safe and tolerable with an initial secondary readout looking at outcomes. And so it's a feasibility pilot that we're starting to develop with SWOG, and we'll be workshopping that today during our 12:30 meeting. So thank you for letting me update you on the sarcoma working group activities. Again, Pacific Room F at 12:30. There's no lunch provided this time. So between the end of this meeting and the 12:30 meeting, grab some food, bring it along. And we'll be going over a little bit more detail of the things that are open and coming in NCTN, as well as discussing the radiation concept in a little bit more detail. And if you're interested in joining the monthly meetings, please just send me an email, davisla@ohsu.edu. And Dr. Wagner and Dr. Harris's contact information is here as well. That's the end of my sarcoma information. I did want to take a couple of minutes, since I think I've got it. Still have some time available to update you as the AYA committee liaison. The AYA committee meeting was yesterday, and we learned about some really lovely AYA leukemia trials that are upcoming, which are both seem pretty relevant for as far as rare cancers. KMT2A rearranged ALL sounds probably not that common. I'm not a leukemia doctor. However, Dr. O'Dwyer, Kristen O'Dwyer from Rochester did an excellent job of making this understandable to a sarcoma doc. And both the S2306 is approved. Really, both of these are highly focused on AYAs. So please let your institution know that these are coming. There are a couple of NCORE trials that are focused on AYAs as well. The kind of immune response in AYA cancer patients to COVID vaccine trials is still enrolling, as is the ECOG trial of AYA PROs. And then for kind of something for everybody to keep in mind for your adolescent and young adult patients, there is a Talk to Me project being kind of championed through SWOG. It's run out of by some investigators at Columbia, and it's really a collaboration between SWOG and SWOG. looking to figure out why in the world our young adults do not enroll on clinical trials. We know that this is a problem. Pediatrics enrolls pretty darn well. The older adults enroll pretty well. Young adults, there's definitely a gap, and maybe that gap of not enrolling on clinical trials has contributed to the much slower improvement in cancer outcomes for this age group. So the project is actually seeking focus group volunteers, is kind of the punchline of me presenting this here. So any patient who is either a current patient or a relatively recent survivor, like, maybe not 10 or 20 years ago, but 5 years ago, 18 to 39 years old, please have them reach out to, I guess you probably have a hard time scanning that little tiny thing, but that's an intake questionnaire. And I suspect that if you Google "Talk to Me AYA trial messaging," you will be able to find some more information. And that is actually the end, and I'm early. Any questions?
Well, thank you for doing such a good job with the working group. I don't have any specific questions, but if there's anybody in the audience that does.
Uh-oh, it's Dr. Ryan.
No, I just, Dr. Harris's proposal, speaking from the executive committee, was, as you mentioned, we wanted to ensure there was buy-in. I might have missed it, but start talking about getting kind of intergroup buy-in. I think it'd be important, very important to have the other NCTN groups committed to this.
Absolutely, absolutely. And so certainly NRG has been involved from the beginning. So Dr. Harris has presented multiple times to the NRG group and also to ECOG and to the AYA sarcoma group multiple times, which includes all of the groups, but it's going to be on my high priority secret agenda for the ASCO NCTN meeting, of making sure that this is everybody's fully aware and that their voices are heard and feedback incorporated.
Okay, well, I think, thank you for keeping us on time and ahead of time. And we will move on to the next presentation, which is Dr. David Chen, who is an assistant professor at University of Washington Fred Hutchinson Cancer Center, and is leading one of our trials, which he will speak about.
All right. Good morning, everyone, and I want to thank the committee for having me to discuss this study. So this is SWOG S2012. This is a randomized phase 2/3 trial of first-line platinum etoposide with or without atezolizumab for advanced metastatic, poorly differentiated extrapulmonary neuroendocrine carcinomas. I know it's a mouthful there. And really the major update that I want to present is that we were finally able to get an amendment done for this study to kind of broaden eligibility, which has improved accrual, and we'll talk a little bit about that. So just to give you an overview about neuroendocrine neoplasm. So basically these are cancers of neuroendocrine cells. So these are our hormone producing cells, which are all over our body. So literally these cancers can arise anywhere within the body, but most commonly they often occur in the GI tract. And as a GI oncologist, I end up seeing a lot of these cancers, and then also a decent proportion that occur in the lung. And then there are cases that are unknown primary. And one of the things about neuroendocrine neoplasms is that we have come a long way in terms of how we classify this disease. So I know we have a lot of different terminology, at least in GI. We're trying to create some terminology that's actually meaningful prognostically and also helps inform treatment options. So in a very broad sense for neuroendocrine neoplasms, we have two pieces of information that we get from the pathology. So one is differentiation status, and then the other is the proliferation indice, meaning how quickly the cells are dividing. So from a differentiation standpoint, we classify them as well-differentiated neuroendocrine tumors, or NETs. And then on the other side is poorly differentiated neuroendocrine carcinomas, or NECs, highlighting a more aggressive disease process. And that's where the focus of S2012 is going to be. And in neuroendocrine carcinomas, we actually have two different types of main histologies. That's either small cell or large cell. But why I want to highlight this classification is one of the things as we were designing the study is we want to make sure that we are including the right patient population. And what I mean by that is that I mentioned that there's a group of patients where they have a well-differentiated status, but their Ki-67 is still high. In other words, greater than 20%, but they're often much lower compared to a neuroendocrine carcinoma. So oftentimes neuroendocrine carcinomas, the Ki-67 is 55% or greater. Well-differentiated grade 3s often tend to be lower to that. And why that's important is that from a prognostic standpoint, so what you have here, this was done by a study several years ago. On the black line are neuroendocrine tumor patients. This is their survival. So these are patients who can live many years to often decades. Neuroendocrine carcinoma patients have a very poor survival, often year or less. But the well-differentiated grade 3s, even though their cells are dividing quicker, they often have a prognosis that's intermediate between the two. And then what we've also learned biologically is that these cancers don't have the similar mutations like neuroendocrine carcinomas, and they often don't respond as well to the treatments that we give for neuroendocrine carcinomas. And so because of that, this study is really focusing on this this poorly differentiated neuroendocrine carcinoma group. Now, when we think about how common do we see neuroendocrine carcinomas, this is a rare disease, and obviously why this committee is leading the study. So this was a nice retrospective review of the SEER database done by Dr. Desaria, MD Anderson, done many years ago, where in the top panel A, we're looking at the pie chart. You can see that a lot of the neuroendocrine carcinomas occur in the lung. That's what we refer to as small cell lung cancer. But then of the extrapulmonary cases, meaning all the neuroendocrine carcinomas that occur outside of the lung, in terms of breaking down the chart, you can see about a third are GI. So we end up seeing a lot of these cancers. But on the right, on the pie chart in B, you can see that they occur in other disease sites. So for example, the gynecological tract and GU tract and as well as others. And then on the bottom left is to demonstrate the prevalence of the various small cell versus large cell histology for neuroendocrine carcinomas. So an orange represents small cell, the blue represents large, and we break it down in terms of disease sites, so like GI and lung. And what you can see is that in most of the disease sites, small cell tends to be the predominant histology. With the exception in GI, we actually see a little bit more large cell than we do small cell. And again, we'll talk a little bit about the relevance for S2012. So prior to the study, what is the standard of care basically for neuroendocrine carcinoma? Because this was such a rare disease, it was really hard to design studies for this group. And we actually based a lot of our data from retrospective studies and often extrapolated from small cell lung cancer, which is with the use of platinum etoposide-based chemotherapy. And so for many years, we were based on a retrospective study until in 2022 at ASCO GI, we finally had one prospective study that was done in Asia, looking at platinum etoposide versus platinum irinotecan. And the take home from whatever study that you look at, the survival is very poor. So these are patients who have a survival of 1 year or less. So obviously there's a lot of room for improvement for this very aggressive disease. Now, as we know in lung cancer, we're using a lot of checkpoint inhibitors. And so in terms of reviewing the data, what do we know about the use of checkpoint inhibitors? So when we use single agent PD-1 or PD-L1 inhibitors in small cell lung cancer, this summarizes some of the major studies. You can see regardless of which checkpoint inhibitor you use, response rates are often pretty low. Progression-free survival is just 1 to 2 months, and survival remains very poor. So obviously single checkpoint inhibitor therapy hasn't really made the progress there. And likewise, in extrapulmonary neuroendocrine carcinomas, with what limited data that we have, we also see the very similar thing. And so obviously doing single agent checkpoint inhibitor obviously is not going to make a lot of progress for the disease. However, when we think about combination treatments, could that be an approach? And so that was based off on Empower 133, which was a first-line study for extensive stage small cell lung cancer, which compared newly diagnosed patients with the control of induction of 4 cycles of carboplatin etoposide and placebo. And if you don't progress, we normally just observe these patients until disease progression, versus the experimental arm where atezolizumab is added to induction chemotherapy. And then if they don't progress after 4 cycles, then you get maintenance atezolizumab to try to control the disease until disease progression or unacceptable toxicities. And based on this study, there was actually a survival advantage of actually adding atezolizumab to induction chemo as well as with the maintenance standpoint. And you can see here on the bottom left here with this table, The median overall survival benefit was pretty modest. So it was just 2 months. So 12.3 months with atezo versus 10.3 months with chemo alone. But it's really the separation of the curves over the long period of time. So in other words, there's a greater chance or likelihood that patients could have a longer term survival. And so it was because of this data that the FDA approved atezolizumab for first-line setting in small cell lung cancer in March of 2019. And so that really, provides some rationale to say, well, what about extrapulmonary neuroendocrine carcinomas? And I will say out in the community, many providers may be doing this off-label, but the question is, we actually have no prospective data in terms of what the benefit of adding checkpoint inhibitor for extrapulmonary neuroendocrine carcinomas. And so while it does make sense that that we extrapolate the management from small cell lung cancer, the question I think that remains in the field is this truly the same disease? So while they do share some similar mutations, for example, mutations in TP53 and RB1, there's also some context-specific mutations that might play into the disease biology. So here on the right, I know it's a very busy table, but this was a nice analysis of basically neuroendocrine carcinomas. And here on the top is just showing you that in the blue, these are all the genetic profiles, mutational profiles of small cell lung cancer. The red is large cell neuroendocrine carcinomas. And then we included a few of the GI neuroendocrine carcinomas. And what you can see here is, yes, they do share a lot of common mutations like TP53 and RB1. But if you look at the graphs and look at all the colors, there's actually a lot of different varying mutations. So one is the mutation profiles are not the same. And then the other thing is that, in lung cancer, that's often the disease that occurs in the context of smoking. And so obviously that may play into the role why checkpoint inhibitors might play a role because of mutational burden. But oftentimes in GI, these are all de novo. It's not a context of smoking. And then likewise, for example, other disease sites like GU, they'll have mixed components. So they'll have a neuroendocrine carcinoma component and a prostate adenocarcinoma or transitional component. And so we don't really know how that affects actually the response to treatment. And obviously we need better biomarkers for this as well. And so because of that, that's where the basis of this study came in for SWOG 2012, where essentially this is a first-line study randomizing patients to one of 3 arms. The control arm is 4 cycles of platinum etoposide, followed then by observation if no progression. There are 2 experimental arms. One of the experimental arms is just like in Power 133, where you get 4 cycles of atezolizumab and platinum etoposide chemo. And if you don't progress, you get maintenance atezo. But the NCI really wanted to evaluate prospectively what that benefit of the maintenance atezo arm is. And so that's why there's this 3rd red arm that's included. And we tried to make this eligibility as broad as possible. And initially, when this study was designed, it was restricted to small cell, and I'll talk a little bit about that. And we do allow up to 1 cycle platinum etoposide therapy, which is very important for this group of patients. And so we'll be looking at the survival benefit of both induction and also of the maintenance atezolizumab. And we have some translational biomarker analysis, which I won't get into for the sake of time, but we'll be collaborating with colleagues at MD Anderson, looking at several transcriptional profiles that may predict response to these various treatments. And then really the take home for the amendments. So as I mentioned that originally when the study was designed, it was restricted to small cell only. And part of that, one of the main reasons, there was actually another ECOG study that was including high-grade neuroendocrine cancer patients, but they only enrolled large cell. They didn't enroll small cell. But that study has now closed. And so that opened an opportunity for us to basically broaden the enrollment to both large cell and small cell. And so right now, as long as you have a diagnosis of a high-grade neuroendocrine carcinoma and your Ki-67 is 55% or greater, just to avoid enrolling well-differentiated grade 3s, basically patients are eligible for this. Many sites also commented about some cost issues related to the scans and insurance not covering those. So we also reduced the imaging frequency and as well as other things. And what that has helped with the study is that, so that's S2012 was activated in December of 2021. We had very poor accrual, 3 patients over 1 year. But with the amendment in January 2023, we've actually now, as of today, 11 of the planned 189 patients. So we are starting to see a little bit more pickup. And so hopefully that will continue. And so with that, we're hoping that if you have not considered the study, that you please do consider opening at your site. And with that, it really takes a village to open a very complicated study. So I want to thank everyone that's been involved, including the study team, our study champions, as well as the Early Therapeutics Rare Cancers Committee for supporting study. And with that, that's all I have.
Uh-oh, Dr. Ryan again.
Always a practical question. So it's good to see accruals picking up. The 11 subjects, do we have a breakdown of the primary site? How many are GI versus, and I'm particularly interested in the non-GI ones
Yeah.
because it's over half of them. And are we getting the word out to the practitioners who are treating those?
Yep. So I know our disease liaison, Dr. Burgess, who's part of the GU committee, he's been selling this. I would say on the 11 patients right now, I want to say 1 or 2 are GU. There's actually very little GI. Most of them have been unknown primary. So.
And it might be good to get feedback why we're not getting more patients on. As you know, like small cell is an emergency a lot of the times.
Yeah.
And has there been thoughts of some, and I can't remember, do we have some flexibility? Can they start?
Yes, they can get one.
They get one cycle and then they can still be randomized.
Yep, correct. And that has helped. And I think one of the reasons is a lot of the sites were waiting for this amendment to go through. And so now that the amendments go through, I think they're willing to proceed with startup for the study. So.
Hi, Rafi Nakash from University of Oklahoma. First question is, are you guys taking large cell neuroendocrine carcinomas of the lung also? Maybe I missed that.
Nope, this is an extra pulmonary study.
Okay.
So yeah.
The second question is from a transcriptional standpoint, large cell carcinomas, because amendment that you showed are incorporating large cell also. So there's a gray zone of large cell, which some resemble transcriptionally like small cell and some resemble non-small cell. I'm guessing there's no data for extrapulmonary large cell on that standpoint, but the fact that you're lumping it together, is that going to create some level of complexity when you're trying to understand where the signal is coming from? Because transcriptional regulation is important.
Yeah.
In that perspective.
Yeah, no, excellent question. You know, and again, there's going to be some heterogeneity for this, but like you said, there's absolutely no data. So one, we need the data. And then 2, because we are collecting all of this information prospectively, we'll be able to do subgroup analysis to look at the various responses based on histology and also transcriptional profiles as well.
One comment and one question. The comment first would be, we probably need to manage expectations with accrual, at least over the next 3 months. The reason is the platinum shortage that many sites in the country are experiencing, and it's going to be really tough. You know, we're starting to ration at our own institution, changing AUCs for carbo and making it 3-week to 4-week cycles instead of, anyway. My question is, because it's struggled with accrual, it's picking up now. Is there some thinking now about simplifying the study into a 2-arm design as a backup or backstop, just in case you run into that problem with the DSMC?
Yeah, so I'll tell you, when I originally proposed this study, it was a 2-arm study, but it went back and forth, and it was a sticking point with the NCI that they wanted that 3rd arm. However, with that said, we do recognize that if that second experimental arm without maintenance it shows to be inferior, the phase 2 analysis allows dropping of that arm if we're not really seeing a signal of activity. Yep.
Rad Al-Jumain, University of Oklahoma. I have a question. You presented earlier the poorly differentiated and the well differentiated with the grade. Do you have cutoff for grade like KI 6 to 7?
Yeah, so the general consensus is usually 55% or greater.
So you have that cutoff.
Yeah, we have that cutoff. Yeah.
Okay.
Will differentiated grade 3 are allowed in this study?
No, they're not.
No.
Okay, well,
right,
thank
thank you.
you. And thank you for persisting. And we're hoping now with this amendment that the accrual will continue to pick up. So our next speaker is Dr. Young Chai, who serves as vice chair of our committee and is a translational co-PI on the DART study, and really has done a lot of work to make the translational components happen, and he will present on that.
Thank you. Thank you for that nice introduction. Good to see you all here in person again here in San Francisco. So I'll be presenting on the TM updates for the DART trial. Here are my disclosures. So we'll talk about the objective and we'll give you some brief update on the background. What are the newest updates in the immunotherapy response predictors. We're going to talk about the academic collaboration, including CMAC. My talk will be followed by Dr. Haymaker will be discussing more in detail about the neuroendocrine cohort TM results. So really our DART trial started with seeing in an effort to replicate the success stories seen in other histologies such as melanoma, anal cell, Merkel cell, renal cell, that the combination of PD-1, CTLA-4 resulted in durable responses. So we are really delving into this unknown space of what are the genomic immune landscape of these rare tumors, predictors of response or resistance. We're now looking at into predictors of immune-related toxicities. And we're going to look at it at the level of genomics, transcriptomics, immune cells, and proteomics. So first, it started as part of the NCI match, where this targeted therapy trial was also focusing on rare tumors. So only when they had screened failure in terms of finding targetable, actionable mutations, then they could enroll into DART when they meet the inclusion criteria of rare tumors. So with that, within the NCI match effort, there has been this already built-in TM efforts, as you can see here. And we'll go into the genomic traits. So our hypothesis that the genomic stability and factors such as DNA damage response gene aberrations such as MMR, HRD, or the level of TMB, tumor mutational burden, may correlate with response to immunotherapy, and that may be seen across different tumor types. So this is a nice figure showing the direct correlation between the objective response rate and the median number of coding somatic mutations seen across different histologies. And here is not only the objective response rate, but also the overall survival directly correlating with the level of TMB here as classified according to the percentage of the TMB in their own histology. And the effect was seen both in CTLA-4 and PD-1 therapies. Here is the lung cancer data that showed nivolumab PD-1 inhibitor used in first-line therapy led to differential outcome in terms of PFS. But interestingly, this trial was a failed trial because overall survival did not land in any superiority nivo over chemotherapy. This is a first trial where atezolizumab was given to everyone, fixed dose first line. And when they looked at blood TMB here, you can see there's a dose response between the level of blood TMB and response rate. But however, as you can see, there seems to be some separation according to TMB in PFS, but not in overall survival. So it depends on the context, what you're looking at, and TMB can, by per histology, can be a marker of response in terms of response rate, but in some tumors, it's not the overall survival that will be the adequate endpoint. What's interesting is CRP, it's a very simple host immune or inflammation marker, was what's better correlating with immunotherapy response than TMB, as you can see in the graph at the bottom. So if you look at bladder cancer, yes, there is a correlation between higher TMB resulting in better overall survival. And you can see that chemo versus atezolizumab here, you can see only in high TMB there was an overall survival benefit. So it is really histology dependent. This is a trial in non-small cell lung cancer where everyone got nivo/ipi. You can see that both TMB and PD-L1 were predictive biomarker in terms of PFS and overall response rate. Here is the Checkmate 227. This is the same regimen that DART patient used. This is not the melanoma regimen where you have high dose ipi given 4 weeks apart. This is 1 mg per kg given Q6. And with that regimen, you can see there is a benefit, nivo/ipi versus chemotherapy in both PD-L1 negative and positive, or TMB high or low. So what was interesting is that they were both independent predictors of response. And here is the subgroup analysis that shows that relationship where PD-L1 and TMB were orthogonal in terms of predicting response. So TMB, where are we with this? We have had a hand tumor or tissue agnostic indication for TMB 10 or greater mutation per mega base pair. And this is based on multi-center non-randomized trial Keynote 158. And as you can see, the response rate there recorded was 29%. And these are the histologies that were found in within that trial. And you can see there are tumors such as small cell lung cancer, salivary, thyroid, vulvar cancers. Here is the response rate seen in TMB high versus low. What's interesting, if you have response, even if it's TMB low group, you will have a very durable response, as you can see in the graph in the middle. So in this trial, what we learned, and across different trials, we're seeing the same phenomenon, is that the PD-L1 level and TMB are not related. And as you can see here in this small cell lung cancer trial, which is very different than non-small cell, you do see correlation with objective response and TMB. And in here, what's interesting is that nivo/ipi did correlate with a better overall survival in high TMB group. This is a very interesting observation presented dissecting different tumor groups. The authors dissected tumor groups into category 1, where CD8 concentration in the tissue correlated with TMB, and 2, where no association was seen. And here you can see that in category 2 group, there does not seem to be dose response relationship between the response or overall survival in terms of TMB and response to immunotherapy. And here is the TMB dissected into binomial and continuous variable, where you can see that category 1 and category 2 tumors are very different in terms of showing association between TMB and response rate. Just one quick question is, if you add chemotherapy, is TMB still predictive? The answer is no. This is based on 2 different non-small cell lung cancer trial combining chemo with PD-1 inhibitor, as you can see. This is exome sequencing using the median cutoff of 175. Now we'll move on to specific gene markers. As you can see, would having specific gene mutation confer any resistance to immunotherapy? So far, the JAK1/2 gene mutation or beta microglobulin gene mutation has been linked with resistance to immunotherapy. You can consider this as like a bottleneck. And HLA class 1 or 2 genotype have been linked to differential response to immunotherapy as well. And in renal cell cancer, there's been a report that showed association of loss of function in PBRM1, which is chromatin remodeling gene, has been shown to have better response, and frameshift indel mutation in renal cell carcinoma has been shown to have differential outcome as well. So we're beginning to think that the way the neoantigen is structured will have impact on response to therapy. And this is research done in my group show that if you have frame shift indel versus non-frame shift mutation, that there seems to be better response to immunotherapy. This is a non-small cell lung cancer data. And this is how that frame shift indel correlates with TMB. So the other trait that we looked in side our trial is the PD-L1 gene amplification. So Dr. Kurdrak's group have found that these amplification have been found across different tumor types, about 0.7% in its incident rate. And you could see the response rate could be quite high. And we did, based on this observation, added new cohort inside DART. Unfortunately, there has been pretty low accrual, and a lot of patients were enrolled based on not the genomic amplification, but based on the IHC. So we weren't able to analyze that cohort due to the small sample size. And then we're looking at germline traits. We do believe that rare tumors may have a higher instance of germline mutations, especially in DNA damage response genes. And we are also looking at the MMR deficiency. It is part of our tissue panel. And this is also currently this also has current FDA indication. So we have pembrolizumab approved in 2017, dostarlimab approved in 2021. And interestingly, both drugs have their separate indication in MSI-high endometrial cancers as well. Now, moving on to transcriptomic space, there has been classic transcriptomic signatures such as IPRES or TIDE or Wnt beta-catenin signature or chemokine signatures, and also HLA transcriptomic loss profile have been used to predict response to immunotherapy, especially this one with the combination immunotherapy. Now, this has been incorporated into different clinical trials. This is non-small cell lung cancer IMpower 150 trial that used 3 different gene signature. This one was CXCL9, interferon gamma, and PD-L1 transcriptome signature that was defined as effector T cell high signature. And within that group, there seemed to have amplified differential outcome in a group where atezo was added to chemotherapy doublet with bevacizumab. Here is atezolizumab data also showing the overall survival and PFS benefit in that subgroup with high T effector signature. In melanoma, loss of the chromatin remodeling gene expression has been correlated with better survival here. And now we're moving on to the immune cell trait, tumor infiltrating lymphocytes, activated CD8 T cells, exhausted T cells, T regulatory cells, and many other immune suppressatory antigen presenting cells have been shown to be correlated with differential response. So we are doing multiplex IHC or IF within our CMAC collaboration. And without the staining, and then now there are quite a lot of papers coming out looking at AI powered H&E staining analyzer that just reveals what are the immune hot, cold, or immune excluded tumors. And this is a paper from my group where we looked at how that correlated with differential pathway activation and how in lung cancer we could do a nice classification of these immune phenotypes, and then how that correlated with differential outcome in non-small cell lung cancer patient. And this is patients over 300 patients treated with single agent immunotherapy. And even in this immune cell trait markers, there seems to be independent association with PD-L1. So they're not correlated. So if you add that into into building your prediction model, you can have a better AUC curve if you combine both. And just the presence of TLS structure, just analyzed by the AI-powered H&E stain analysis, also showed better survival outcome in small cell lung cancer patients treated with immunotherapy. So these are the emerging areas I think that will show a promising predictor. So we are planning to analyze that in our DART cohort as well. So moving on to protein markers, yes, we stained our tissue samples with DACO28-8 immunohistochemistry. This was companion biomarker for nivolumab. And we do know that in renal cell melanoma that this PD-L1 marker did correlate with better survival in nivo-ipi treated group. And we are looking into blood protein markers. So the mass spec, the MALDI-based proteomic signature has been shown to differentiate patients treated with either single agent immunotherapy or combo immunotherapy, nivo-ipi, in both lung cancer and melanoma, as you can see here. There are other papers coming out that are using now multiplex ELISA antibody assay, measuring 1,000 plasma proteins, and they're coming up with differential plasma protein signatures that can predict response to immunotherapy only treated group. And this is a non-small cell lung cancer paper. So now we are really looking into now serum proteomics, because when you're talking about blood protein, then you are 99% of those proteins are coming from host. And of course, we're looking into germline mutation. We're looking into clinical variables. We're looking into lymphocyte neutrophil ratio as well. And this is now pretty much well known that absolute neutrophil count and platelet count can, at baseline, at start of immunotherapy, may correlate with differential outcome. And neutrophils' predictive power is not only confined to immunotherapy, but across different chemotherapy regimen, as you can see here. But the best data comes from the neutrophil to lymphocyte ratio, as seen in different immunotherapy-treated clinical trials. So we are looking into that in our DART cohorts, and there seems to be dose response relationship with that ratio, but also independent relationship with TMB as well. So they could all be orthogonal, build upon like a prediction model type of vectors. So I think we really need to understand, how do we integrate these data? What are orthogonal and what are bottleneck? Just like I said, the JAK1/2 mutation doesn't matter if you have high PD-L1 or high TMB or high transcriptomic risk factors. You can't turn on the interferon gamma pathway because of the bottleneck, then you're not going to get anywhere. So understanding the flow, the structure of these predictor is, I think, very important. But at the end, I believe AI can help us figure out where the right weight should be applied. So the papers are coming out looking at all different levels, like an immune cell, PD-L1, transcriptomics, and they, as you can see, what's the main message is they're orthogonal, they're independent of each other, they're not correlated. And this is another paper showing the same lesson in melanoma. And in their transcriptomics, they looked at PD-L1, CD8, LAG3, and STAT1, and still they were independent of each other. So just a lesson learned from LungMet study in our own lung committee here in SWOG, that in this cohort where nivo-ipi versus nivo was randomized, because of the sample size, we weren't able to get to any conclusion. But it's interesting to see that no benefit with nivo-ipi over nivo were found, but exploratory subgroup analysis found that there's a potential OS benefit of nivo-ipi over nivo in patients with TMB high and PD-L1 low. And here was the 5% cutoff. And this is the left side graph. You can see PD-L1 negative but the nivo-ipi had the highest benefit in TDL1, TMB high group. So I think that's one lesson we learned from our SWOG trial here. And now we're looking into the immune-related toxicities. Of course, there's been papers alluding into better responses are seen maybe with patients who experienced immunotoxicity, and those data are coming out. And those it's a series. Those with the high will still be alive with the and having durable response are the ones who experience immune-related adverse events and had stopped the therapy. And it's really interesting to see that in that Checkmate 227, where the same regimen nivo-ipi was used, that median overall survival 4-year was 44%, regardless of the PDL1 level. And these are the, remember, they came off the study probably during the first or second year, and they're still alive at 4-year, about 44%. So this is quite alarming and a promising data, I guess. And my group has looked into, can serum proteomic predict immune-related adverse events, and there seems to be some signature. And my group and other groups are looking into that. And I think DART is also a very good platform to look into the immune toxicity predictors, because we have collected the serum and we can do the MALDI-based proteomics analysis. So again, what are the positive predictive factors? What are the negative? What are the bottlenecks? And understanding that at pan-tumor level, we do think it's very important. And then also getting down to the clonal structure. What is the best way to assess heterogeneity index? Because it's just not the neoantigen burden, but understanding whether it's clonal or subclonal does impact the outcome of the long-term survival. So in DART, we've collected pretreatment tissue as well as blood. It was at baseline, at first scan, and scan at progression. So we had collected 3 time points blood markers, and these are the initial biomarkers that were designed. And we have PDL1 immune biomarkers, germline proteomic signature, ctDNA, and tumor next-gen sequencing. And we have industry partners, which are Circulogen and Biodesics. And this is because we've closed the new enrollment. We are sending all our SWOG biorepository bank samples to these companies. And then we initially started with the JAX collaboration, but moved on to CMAC because they just launched the pilot cohort at the time when we were designing the TM portion of our study. And these are the assays platforms done through CMAC. And Dr. Haymaker will get into more details. And these are the biomarkers that are currently being done, mostly in MD Anderson, but as you can see, Olink and Mount Sinai, CyTOF at Stanford, and then all the data analysis will go to the CIDC, Cancer Immunologic Data Commons at Dana-Farber Cancer Institute. So we'll be really dissecting the data at multi-omic level. And these are the immune profiling workflow. And at first we were very ambitious, trying to, we're going to call this rare tumor TCGA, try to understand what's going on. But then, of course, there's a lack of funding and limited resource. So we had to prioritize our group. So this is how we got 3 layers, tiers. The first tier group would be the group with the most response seen. And we first delved into the 3 neuroendocrine tumor cohorts. And then we found groups with at least 2 patients with objective response, and then went down to at least 1 patient with response. And currently, we've finished the neuroendocrine 3 different cohorts. And the things we've looked at are PDL1, multiplex IF panel, nanostring, Olink, and then whole exome sequencing. And we've just finished the cohort 26, which is a thyroid cancer cohort. And we are just wrapping up the analysis, writing up the paper. This would be the first paper to come out with the clinical and translational piece all together, because prior papers all been just clinical piece only. And then our next plan is to move on to metaplastic breast cancer and angiosarcoma group. But obviously, we are open to looking for more funding sources. We're looking to partner with other commercial biomarker companies to get into the details of other rare tumors. Here are the cohorts that we are initially looking into with the limited budget that we have. And neuroendocrine cohort will be, the update will be given by Dr. Haymaker, but here is the brief update for you here, audience here, for the thyroid cohort. So what's really interesting is, so we have 19 patients with objective response rate of 21% in thyroid cancer cohort treated with nivo/ipi, 4 patients with partial response, 1 patient with unconfirmed partial response. When we got the tissue, we lost 2 patients. So PD-L1 level was able to be done in 17 patients. When we get to the whole exome level, because of the tissue insufficiency, we were only able to do 9 patients, but we were able to still retain 3 patients with PR or unconfirmed PR. So this is the PD-L1 data. Surprisingly, all of them, I mean, we know a lot of thyroid cancer patients have cold tumors. The PD-L1 level was very low, but even with that, if we separate them by positive or negative, as you can see, you don't really see much separation in the curve. Here, the upper curve is when they have response. Did that translate into overall survival benefit? The answer is yes. The lower curves are showing by TMB cutoff with median. Do you see any differential survival outcome in terms of PFS and OS? The answer is no. We looked at BRAF, RAT, copy number variation burden, absolute neutrophil count, platelet count. We did not find any meaningful correlation there, but at least we've done our job to look into our TM portion, the risk factors. So the consistent findings across different cohorts, neuroendocrine, thyroid, and pancreatic neuroendocrine was that the PD-L1 expression level was not correlating with response or survival. There was no clear association with circulating ANC level with survival, but there was some PFS trend seen in pancreatic neuroendocrine cohort and overall survival trend seen in thyroid cohort. Platelet count did not, knowing that it is an immune suppressive factor, was not correlating with any differential survival, at least within the DART cohort that was analyzed. So with that update, I'd like to thank everyone involved in this comprehensive effort, and it's really been a team effort. I'd like to, thank all our colleagues, especially, now with Dr. Haymaker's team on board. It's been a pleasure to work with them, exploring the data together. And especially now that the trial has closed new accrual, where we now will be really focusing on looking into different association with different biomarkers entering the field. So thank you.
Are there any questions?
Huge kudos. This is such a Herculean effort. I don't know of any other cooperative group study that does such an extensive translational effort. So huge thank you for showing us the way. And we'd love to learn from you in our iCheckIt study that's focused, big cohort studies focused on IRE prediction, where we have longitudinal blood collection as well, germline, as well as whole blood. along the way in plasma, and also baseline tumor, and now also longitudinal microbiome that's being added on. So, your experience, how you've been able to leverage these various resources to also help with really processing the specimens and really getting the biology data would be fantastic. Maybe just two aspects. I'm coming from the biomarker world and sit on the breast cancer ASCO biomarker guidelines. And as we've breast cancer has been fortunate early on to add biomarkers. And I think there the message was, and the learning experience was in these, how do we incorporate these various biomarkers? It's what's feasible and what adds clinical value, right? What adds clinical validity, and particularly also showing clinical utility, that adding another biomarker, that's another costly undertaking to that clinical scenario. And does it really improve outcomes for that patient? Does it improve our patient selection process? I think that also will probably be the guiding principle here. But at the same time, you're figuring out biology. So as you're going broad, so big kudos.
Thank you. Thank you. So totally agree. Feasibility, practicality, and the more biomarkers you have, if it's a tissue biomarker, you're going to exhaust tissue. As you can see in the whole exome, we lost more than half of patients because of the tissue insufficiency. So I think we just need to incorporate the biomarkers that are not, it's really coming from different angle. Like if you have tissue, maybe blood, phenomics is important. And we're doing H&E slide anyway. So if we can use that, the image data, and then I didn't talk about radiomics, but it's something that we have in mind that we'll be looking into. Radiomics can be predictor of pneumonitis too, as well as a response to immunotherapy. It's still controversial because of so many confounders, but I think, looking at different biomarkers at different level and integrating that using AI platform is definitely going to be the future.
Okay, thank you so much for what is really one of the most important efforts in DART, and that is the biomarker effort. And on that line, Dr. Kara Haymaker, who has been doing some of the analysis and always presents with huge enthusiasm, which I love, is going to talk to us about some of the translational findings from the neuroendocrine cohort.
All right, good morning, everyone. I'm so excited to be able to join you today and provide some updates on our findings for the non-pancreatic neuroendocrine DART cohorts. And thank you so much for the wonderful collaboration between the SWOG team and NCMAC and also NCI Match. So just some unaffiliated disclosures. So like I mentioned, this study has taken a village. So not only all of the amazing efforts led by the study leads that you can see listed here, this also involves tissue that was provided by the NCI Match study. The CMAC, we're working with 3 different CMACs. This is led by the MD Anderson CMAC, but our Mount Sinai colleagues, as well as our Stanford colleagues, are contributing analyses and interactions. And you can see some of those leads highlighted here in red. And then also our amazing support from the NCI. So how are we actually trying to break down in an exploratory analysis manner what is going on in the tumor microenvironment, as well as changes that we can assess longitudinally using blood that's been collected over time from the patients in this study? So what we are doing right now and what I'm presenting today is going to be involving some gene expression profiling, future tumor mutation burden, and Dr. Chai has highlighted nicely the importance of this within the microenvironment. Also, in looking at the PD-L1 chromogenic IHC and the now we understand lack of correlation across the cohort so far, but really trying to understand how the cells are interacting with each other. Do we see ligation of the PD-1, PD-L1 axis? How are the cells actually interacting within the microenvironment and clustering together? And these are going to be some fun updates that we'll talk about today. And then when we consider what happens over time, for this study, we were able to access longitudinal blood that was collected at baseline, also at cycle 2, week 9, and then again at progression. And this allowed us to understand changes in circulating biomarkers. So we utilized a profile platform called Olink, which allows us to look at cytokines, chemokines, and other soluble receptor changes in the circulation. So we can look at immune activation states. At the cellular level, we applied a CyTOF technology, which allows us to look at the actual cells themselves and their phenotype changes over time, to really try to understand how we might be modulating the circulating immune microenvironment with this combination approach. Now, as was mentioned by Dr. Chai earlier, we are working with 6 different cohorts, and I'm going to be updating on 23 and 52. These are the non-pancreatic neuroendocrine cohorts. And just as a reminder, cohort 23 is a mix of grades, but based upon the clinical response that was seen in this cohort, cohort 52 is established to specifically focus on high-grade non-pancreatic neuroendocrine cohorts. And so we've been able to combine these data sets together to try to understand what's happening in this study. In terms of what we had available for this type of analysis, you can see the upset plot that is shown here. Overall, I'm going to be updating on most of the assays that we've been able to utilize to date to understand the microenvironment. And we've been able to access data points from a total of 46 patients and a total of 43 that had tumor tissue available for analysis. We did have a few cases that were dropped out due to QC issues, but you can see overall across all of the assays that we've been able to apply thus far, we had 15 patients that had everything that was able to be analyzed. Now, when we look at this, this data set in the sense of what we had available for analysis and the associations with overall survival and progression-free survival, you can see that across the combined cohorts, we had a median overall survival of 8.9 months and a median PFS of 3.6. So if we first focus on the tissue, trying to understand at baseline, do we see biomarkers that could be associated with subsequent response to this combination therapy? As was previously discussed in the fall meeting last year and also just by Dr. Chai, is that we don't see any association of PD-L1 by chromogenic IHC with response. In fact, in this combination cohorts 23 and 52, we had very low PD-L1 expression. Only a few cases were positive. You can see an example of a positive case that is shown here in B, and we saw no association with PD-L1 expression in terms of response, PFS, or OS. And you can see the OS in C and the PFS in D. Now, what we wanted to then do is say, okay, well, if we don't see PD-L1 associating with outcomes, what about the transcriptomic profile within this microenvironment? And so to assess that, we utilized a nanostring. This technology was necessary for us because of the quality and concentration of RNA able to be co-derived from this tumor tissue. And what we found, interestingly, when we look at the overall profile, is that we saw this, this TNF receptor family member that was strikingly associated with high-grade lesions. And this is known as the B-cell activating factor receptor. And you can see that on the very top line of this heat map that is shown here. We also saw other gene expression profiles that were more related to the actual grade of the tumor. And so we saw differential CDK, tubulin, different types of proliferation markers that were associated with high grade versus the low grade or intermediate counterparts. Now that made us ask the question of, well, does that mean that there are actually B cells in these tumor microenvironment? And in fact, when we utilized an algorithm called Timer to deconvolute the gene expression profile within the microenvironment, we saw that B cells were predicted to be present, and not just present, but actually to associate with response. Here in A, you're seeing progressive disease patients, stable disease of less than 6 months, or patients that had a CR/PR or stable disease of greater than 6 months. And we saw that really a B-cell association was highly significant relative to a positive outcome. Interestingly, we also saw there was kind of a stair-step approach in terms of the other antigen-presenting cells that were present, such as dendritic cells. We saw that DCs were the highest predicted in patients that had a good outcome. And in stable disease of less than 6 months, there was kind of this intermediate presence within the lowest presence in progressive disease. When we looked at the B-cell association with progression and overall survival, we also saw that patients that had a higher B cell score at baseline did better in terms of their survival. In addition to that, as you can imagine, we saw a higher scoring in terms of antigen processing and presentation. And we also thought, well, maybe these B cells could be involved because we have a tertiary lymphoid structure or immune aggregate formation. We know in other types of tumors, especially melanoma, non-small cell lung cancer, very inflamed tumors, the presence of a tertiary lymphoid structure is a positive association with subsequent response to IO. And in addition to that, it can also come up with response to IO. And so we applied a signature deconvolution to look for a tertiary lymphoid signature score in the nanostring data. And we saw that this was higher in patients that had a better outcome. And you can see the specific score that's shown down here on the bottom. But, you know, that is just a score. We wanted to really understand with our eyes. We have a really strong translational pathology component in our CMAC, and we wanted to understand if we could really identify plasma cells or immune aggregates within this tumor microenvironment. So not just a predicted score, but can we quantify it, right? And then see if that can be associated with actual outcome. And so what we did is we went back to the H&Es, and we did scoring for plasma cells, as well as looking for the presence of tumor and lymphoid aggregates. Now, our translational pathology colleagues here, this is Dr. Luisa Solis Soto and our postdoc, Caddy. And what they did is they utilized an approach where they defined a lymphoid aggregate as a cluster of 50 or more lymphoid cells. If they had a germinal center or not, then they could be classified as a mature tertiary lymphoid structure. In addition, when we went back to the H&Es, we also saw that there were actual plasma cells present. I am not a pathologist. I didn't realize how you could score plasma cells within an H&E. And it turns out there's a very robust, very clear visual of how a plasma cell looks within the microenvironment, and we were able to assess that. Now, when we broke this down and looked to see within the tumor tissue, do we have plasma cells associated with lymphoid aggregates? The answer is sometimes, but not always. What we're showing here in this table is if we have lymphoid aggregates present, you can see this blue box. In 5 cases, we also have plasma cells, but in 1 case, we do not. And furthermore, when we subcategorize this based upon response, we do see that our high-grade responding patients do have a slight correlation with the presence of a lymphoid aggregate, but not all of them have a lymphoid aggregate. And so this is a really interesting view of the potential role of B cells within the microenvironment in these non-pancreatic neuroendocrine cohorts. So then we asked the question, okay, well, if we can identify these B cells, identify these plasma cells, does this actually associate with response and outcome? And so we broke this down into lymphoid aggregate positive tumors versus lymphoid aggregate negative tumors. And what we were happy to see is that our scoring system nicely recapitulated what we saw by eye. So if we predicted that there were B cells present within the microenvironment, we could actually see those with our eye. And this was able to be, in some cases, associated with clustering and a lymphoid aggregation. In addition to that, when we look at our TLS signature, we also saw this was consistent with the presence of an actual lymphoid aggregate detected by our translational pathology approach. But it was not always just patients that had a positive outcome. What you can see here in patients that are responding, they are the little squares. And what we found is that, in fact, while most of the patients that responded did have a lymphoid aggregate, there was 1 patient that did not. So our stats are not perfect. And this is always the caveat of having a small number of patients in an exploratory study. But overall, we do see that there is a real strong association with lymphoid aggregates, B cells, a tertiary lymphoid structure, and subsequent response in these high-grade non-pancreatic neuroendocrine tumors. So then how are these cells actually interacting with each other? Can we understand whether or not that the presence of cytotoxic T cells, the engagement with the tumor microenvironment is happening along with the presence of B cells that we already now have clearly seen to associate with response and outcomes? So in order to do this, we utilized an assay called multiplex immunofluorescence, and this is led by Dr. Edwin Paracuentas on the team and analysis by Zhixin Zhang. And what we saw is that, like we saw with chromogenic PD-L1, when we look at PD-L1 using this other type of technology, it's very low, and we don't see any association with response. What we did see, however, is that we found that tumor-infiltrating lymphocytes that were detected both in the stroma and in the tumor regions did express PD-1. And we also saw and you can see those in the purple box, we also saw a very high presence of memory CD4 T cells. And this is something that was very interesting because in most immunologically hot tumors, unlike what we have here in our nets, we see an association more with memory CD8s. But here in this setting, the dominant T cell population is actually CD4. In addition to that, again, we also see cells present both within the stroma and the tumoral region. So we asked the question, well, could this then associate with grade and response? And what we saw is that when we look at the overall just presence or immunologic hotness, based upon the cell types we can quantify with this assay, does that associate with grade? The answer is no. We have low and intermediate grade tumors that have just as many immune cells as high grade tumors. And furthermore, when we look at the high grade tumors, we can see that this does include cases where we have lots of TIL, relatively speaking, and cases where we don't have TIL. But if you do remember, we have B cells. So there seems to be a myeloid component or a B cell component that is there at baseline. And even if there is not a T cell presence detected. So then we wanted to understand that a little bit more and try to understand how these cells could really be interacting with each other. Just because the overall frequency of a cell type is the same does not mean that they're interacting and clustering in the microenvironment in the same way. You could have suppressive cells that are clustering more around the edge of the tumor versus within the tumor itself. And you can have direct interactions with your myeloid compartment and your tumor cells within the immune side as well. And so in order to address this question, what we did is we utilized a spatial analysis approach that's led by Dr. Gayatri Kumar on the team. And what we're doing here is we're looking at the H&Es, the multiplex immunofluorescence, and the gene expression profiling together. And then utilizing point pattern analysis to identify the specific cell types, we are looking at clusters of 20 cells or more that have to be within 25 microns of each other. So not that they're far apart from each other. We're really wanting to take a deep dive and understand how they're literally interacting with each other. And if that is something that discriminates between patients that have a good outcome, even though they might have a similar frequency of cells as non-responders. And then we can also look at different types of clustering algorithms to see how they're interacting. And so I'm going to run through this fairly quickly. This is very fresh data, but just to show you our thought process in terms of how we're trying to take a deep dive into understanding the microenvironment in this context. So here you can see the H&Es. You can see we have plasma cells in this high-grade responding lesion. We also have tumor cells, and we have some immune aggregates. When we look at the multiplex immunofluorescence, we can see those cell types. Now, when we take the cell segmentation information and we plot the cell types here, each dot is going to represent a different type of, is a cell, and it represents a different type of phenotype. So we have our CD4 T cells in black. You can see that they are the dominant population of T cell present within the microenvironment. When we apply clustering algorithms, we can see that we have a number of different clusters that are present, and then we have other cells that are not associated with any cluster. And the point I really want to make with this slide is what's interesting is that our T helper cell populations are the highest frequency cell type defined. This is not necessarily a cytotoxic T cell association. There's really something going on here with the T helper cells that are coming into this tumor microenvironment. In addition to that, we also see in patients that have a good outcome, we have an interesting clustering of T regulatory cells. The T regulatory cells that you can see here in blue in the dot plot right here, they're actually clustering with the other types of T helper cells. They are not on the periphery. They seem to be engaging with the other cells within the microenvironment. Now, on the other hand, when we look at patients that, again, have the same number of immune cells present, but don't respond, these patients have a slightly different profile. In this context, we actually see that there are more cytotoxic T cells that are present at baseline than the T helper cell component. And you can see those in green here in the clustering. In addition to that, we see that there are Tregs to the same degree, but they don't behave in the same way. Instead of actually interacting with the other CD4 T helper cell populations, they appear to be more on the periphery. So you can see those in blue. They are actually hanging out around the outside and not interacting with the T helper cell lineages. So suggesting that there could be some different mechanisms behind their suppressive function that could associate with subsequent response. Okay. So moving on to what we see in circulation, we really wanted to utilize the blood component to try to understand if we could be identifying potential immunogenic priming. This combination, of course, is associated with anti-CTLA-4. We know that when you are blocking CTLA-4, you can restore priming events. And because the tissue is only available at baseline, we're not able to look at that directly in the tissue. But using the blood, we are able to see if we are getting immune activation and could we actually be expanding new T cell populations. And to us, this makes a lot of sense because, again, if we have cases where patients are responding, but they don't have T cells at baseline, maybe we're bringing them in with this combination, and that's really what's happening. And so to address these points, we utilized the O-Link system with Mount Sinai CMAC to look at immune activation in circulation in the plasma. And indeed, what we see is that here in patients that or actually irrespective of response, when we look at cycle 2, week 9, or at time of progression, we see that there are inductions of different types of pro and anti-inflammatory factors. In addition to that, when we break this down by response in C, now we can see in red different proteins that go up in non-responders, and we can see in blue different proteins that go up in responders. In fact, when we look early, cycle 2, week 9, we can already see that interferon gamma is coming up in patients that have a good outcome. IL-13 is also coming up in patients that have a good outcome. On the other hand, different types of other factors such as IL-8 are actually coming up in patients that have a worse outcome. So we do see that this combination is promoting immune mobilization and activation in patients that do better. But what happens with the cells? Are we again promoting activation of different cell types in circulation? And so utilizing a CyTOF-based approach with our colleagues at the Stanford CMAC, we looked over time at the different cell changes in their phenotypic profiles and activation states between baseline and week 9. Now, when we just look at baseline, what we can see from the very beginning is that in the circulating microenvironment, we have different steps of or different presence of effector memory cells that are CD4 in origin. You can see that is shown here at the top and at the bottom. We have 2 different effector memory CD4 lineages that are higher at baseline in patients that have a better outcome. And you can see there are various clustering associations here on the right. And beyond that, when we look to see how they change over time, we see that not only are the CD4 effector memory cells expanding in patients that have a good outcome. We also see that PD-1 and different types of memory cells that are CD8 in origin are also coming up. So it does look like we are potentially reinvigorating the immune response, but this does not definitively prove that we are priming a new T cell response. So in terms of what have we learned and then where are we going? What we have found so far looking in the tumor microenvironment is that the presence of B cells and dendritic cells at baseline prior to therapy correlates with improved outcome. We don't have mature tertiary lymphoid structures, but we have very clear associations with lymphoid aggregates that we are defining as 50 immune cells. These are very present in both responders and non-responders, but the majority of the responders have these lymphoid aggregates present, and there's a tertiary lymphoid structure signature identified at the transcriptomic level. We do not see any association of PD-L1 with outcome. We don't see that grade is driving immune infiltration of T cells, and we see that responses can be identified in very immunologically cold microenvironments as well as moderately infiltrated tumors. And there's also this very high degree of heterogeneity within the cellular neighborhoods, but we have a very clear presence of CD4 T helper cell populations. And there is a potential for a differential type of organization of these cells within the microenvironment that is associated with their the subsequent outcome of the patient. And this relates more to T regulatory cells and also sometimes to cytotoxic T cells. And in circulation, we see that there are very specific types of effector memory cells that are coming up and enriched in responding patients. And we see that there is an association with immune mobilization and activation, looking at soluble cytokines and chemokines. Now, in terms of where we're going and taking this further, now we want to understand the genomic contributions to these profiles. So we're looking at TMB, CNV, HLA loss of heterozygosity, as well as other types of mutations, JAK-STAT pathway mutations, et cetera. The other point here that we've been trying to address is immunologic priming actually associated with outcome? Because again, if you have a paucity of T cells, why are these patients responding? And that's going to be a critical an assay. And what we're doing to address that question is we're utilizing TCR sequencing of both the tissue as well as the circulome over time. And hopefully I'll be able to share that with you guys in the near future. And then finally, given the really strong presence of CD4 T helper cells that are not necessarily Tregs within the microenvironment, we're trying to utilize our gene expression profiling to deconvolute these different T helper cell lineages to see if we can better refine our understanding of different profiles of those cells that could contribute to an anti versus pro-tumor response. So thank you all for your time and attention. I would be happy to take any questions. And thank you again for the organizers' invitation to present our findings today.
We're running over time, so I'm going to give it one question.
Thank you. Really nice talk. My questions are about myeloid-derived suppressive cells.
Yes.
If you've had a chance to look at like the M1, M2 polarization and what that looks like in these nets.
Absolutely. So at the cell level, the multiplex immunofluorescence panels don't allow us to distinguish the M1, M2 phenotype. But what I can say is that the myeloid component here is very low. On the gene expression profiling level, we do not see an M1, M2 score differential. So far, it doesn't appear that there is evidence that that is what is happening. We also don't see that the myeloid compartment is clustering in any different way between responders and non-responders. Really, it seems to be more about the B cell signature. Okay. Thank you all so much.
Well, thank you. And our next presenter is going to be Dr. Sandeep Patel, who has really led the DART trial from his inception as a young investigator. I heard recently he's not a young investigator anymore.
Grays give it a
Yeah, so he's still young to me, but he's
exactly right next to you.
Yeah, but really done a great job with the DART trial, and looking forward to your presentation.
I'm looking forward to the presentation too. I just got to get to the right slide deck. All right, thank you, everybody. Great. So I guess bench to bedside to back. And then so I'll be going over the clinical aspects of S1609 DART and really the genesis on what led to the really interesting translational data you heard from my colleagues. And so really this came up as an idea. And one of the questions back when we were first thinking about this study back in 2015 was a lot of patients were asking, well, what about immune checkpoint blockade for my cancer? And oftentimes these patients will come into an experimental therapeutics clinic because they're more than 3 syllables in the name of the type of cancer they have, and they don't neatly fall into, lung or breast or GI. And it turns out it's a quarter of patients. This is EU data, but if you look at the US data, it's very similar. Almost a quarter of cancers are quote unquote rare. And more importantly, it's a disproportionate burden on patients who are younger. In fact, up to age 40, quote unquote, rare cancers are more frequent than the common cancers that we talk about. And so historically, it's been difficult to do clinical trials for this population because of the myriad of different organ types that really cut across the way cancer centers and most clinical enterprises are structured, right? The hematologic malignancies, you walk through one door, gynecological malignancies, a different one, head and neck, a different one. And then if you look in particular, when we're talking about neuroendocrine cancer, it's even more complicated, right? Because you get neuroendocrine tumors arising in the lung, which are very different than those arising in the GI tract and the gynecologic tract. And so it gets very complicated. And so what this has really led to is kind of this historical inertia against doing clinical trials and this tautology that, well, we can't do clinical trials in rare tumors because they don't accrue. Well, why don't they accrue? Well, we don't have any clinical trials open for patients with rare cancers. And so we sought to address this through the construct of S1609 DART. This is kind of a snapshot of what we've seen, and this was a nice feature in the cancer letter, which when you hear you're in the cancer letter, you always go quickly to the website to make sure it's not anything on the wrong end of the street. And so we started with 33 cohorts in 2017, and we ended up with 53 cohorts more recently, which I'll walk through. And this is really a nice dashboard that Megan Othis and others helped create, because the study accrual was so drastic that the ability to allocate slots for patients really came into question. In fact, we created a drug supply issue for NCI-CTEP in 2018 related to the accrual rate for the study, which is a good problem to have, I guess, relatively speaking. And so let's talk a little more about the genesis of this study. We know why 2 immune checkpoint inhibitors, why not 1, why is continuous dosing of ipilimumab as opposed to induction dosing like they do in melanoma or renal cell cancer. And so one point, and these are 2 studies led by SWOG institutions, is that we've seen efficacy with anti-PD-1 directed therapy in various rare cancers, whether it be Merkel cell carcinoma or anal cancer. These are both led by SWOG investigators at SWOG institutions. And the idea that combinatorial immune checkpoint blockade, if we're looking at a signal finding study, we may only get 1 chance to look at amelioblastoma in a study. And so why not try to maximize our ability to find a signal through dual immune checkpoint blockade, though with the idea of maximizing patient safety, and in so doing, picking a lower dose of ipilimumab, 1 milligram per kilogram every 6 weeks, as opposed to the induction dosing, for example, that we use in melanoma, 3 mg per kg times 4, for example. And so this is the first NCI-funded rare tumor immunotherapy basket study. It's now close to accrual, but it's been quite the run, and I'll show some of the highlights. The primary objective is objective response rate, a signal finding study with a Simon 2-stage design, 6 patients in the first cohort and then 10 subsequently, and the cohort's considered clinically positive if there are 2 or more responses. Imaging assessments were performed every 2 months initially and then could be spaced out. And as mentioned, we tried to Goldilocks the dosing of dual immune checkpoint blockade in terms of signal finding, but also trying to, in terms of maximizing objective response rate, but also trying to minimize toxicity for patients with a variety of tumors for which this regimen had not been tested before. And so this is the dashboard with the most up-to-date statistics. You can access it as readily as I can. And as I put together these slides last week, we had 798 accruals across 53 cohorts. And so let's talk about some of the cohorts of clinical interest. Some of these you may have heard about at meetings, but to highlight this into a single talk. So neuroendocrine neoplasms themselves are a heterogeneous group of rare tumors. They can occur throughout the body. We heard about this from my colleagues earlier. One issue is many are well-differentiated low-grade tumors, very indolent biology. These are often the patients who do really well on a somatostatin-directed therapy, whether medical or radiologic, so to speak. And then poorly differentiated high-grade tumors, which really recapitulate small cell cancer. This is lung, GI cancers, unknown primary, which happens all too often as well. And these are classified in a very complex histologic classification scheme that's actually been updated at least once during the conduct of this study from a pathologic standpoint. And pancreatic, of course, is classified as a different neuroendocrine tumor aside from gastrointestinal, and then we have lung and thymic. And so prior studies, and this was highlighted earlier, of immune checkpoint blockade monotherapy showed a response rate about 5% in high-grade neuroendocrine cancer. So in this cohort, we had 32 patients, and this did not include pancreatic neuroendocrine, which actually is a different cohort based on its biology. And 56% of patients had high-grade carcinoma. Half these patients had GI as a primary, and median number of prior lines of therapy was 2. When we look at the demographics, this is a population that had a wide range of ages, right? 36 being the youngest, 81 being the oldest. But we look at the primary sites, and this is kind of one of the issues with these kinds of neuroendocrine directed studies is it's really hard to splice the atom and figure out where these primaries are coming from. And oftentimes you don't know, it's your best guess in clinic, right? That there's nothing on the PET scan that has an arrow pointing to, hey, the tumor started here, right? And so you're just making your best guess clinically. Half of these patients are high grade. These patients had high-grade disease, and about a third had intermediate grade. And you can see anywhere from up to 7 lines of therapy, likely for the lower grade tumors, patients with lower grade tumors. And so initially, when we looked at this data, this is what the waterfall plot looked like. And it was confusing, and we looked at this together. The suspected primary organ of origin did not delineate which patients responded or not, as you can see here clearly. But then we looked at grade, and by grade, you really see a bimodal distribution, right? You see patients with these deep responses, often heavily pretreated, and you also see patients who didn't get benefit, and they unfortunately continue to progress, right? Because for those group of patients, giving this immune checkpoint blockade combination was probably no different than giving normal saline, but for the patients who did really well, it was life-changing. In fact, just yesterday, I actually saw one of the patients, and it's really interesting, just as an aside, to see a patient in person who's been on your study and know which bar they correspond to, right, in this data. But it really goes back to the work that everyone in this room does, and my colleagues have done, and that, we all look at data, but these are all patients, and it was just really heartening to see the number of weddings and graduations this patient's been able to go to. And so when we show this data, it just really brings home when we talk about these patients and you get to see them. And so that patient was one of these bars, who is the arrow continuing. He's been on therapy over 5 years now. And it just really highlights that when you get these nice durable responses, these patients can do well for a long period of time. That patient, just to finish that story, had been on 5 prior lines of therapy, had been given 6 months, basically, when he went on the study. And now it's going to be 6 years, hopefully, ongoing. And so summarizing this data, the objective response rate, 44% of patients had a response. The retrospective view of this data, right, because we didn't know a priori going into this that high grade would have benefit, though I'll get into that part of the story momentarily. So looking at this high grade group as a subgroup in a retrospective fashion, pulmonary, about 2/3 of patients responded. The objective response rate in high grade neuroendocrine carcinoma was about 40%. In GI, it was about 1/4, though, as mentioned, the primary organ of origin did not seem to delineate or discriminate between responders and non-responders. And you see here a durable benefit in this subset of patients with response. Anytime you're trying a new regimen, or at least a new regimen for this patient population, toxicity is very important. And so fatigue and nausea are the most common, most likely related to their underlying cancer. Their most frequent immune-related, treatment-related toxicity is really related to thyroid dysfunction and liver dysfunction. There's one case of high-grade colitis, but no cases of pneumonitis, and thankfully no grade 5 toxicities. And so limitations, we did not have central pathology review. These are local pathology reports with mitotic rate and Ki-67 utilized from the reports that are provided to us. We heard some of the excellent translational data that has been generated and is being analyzed. And then it was local adjudication of organ of origin, though at least based on our first glance at the waterfall plot, that really didn't seem to drive the decision-making in terms of, or the outcome in terms of which patients responded versus not. This wasn't a pre-specified analysis for this cohort. We discovered that it was just the high-grade patients, high-grade neuroendocrine patients that are benefiting. And it's a single-arm cohort. It was non-randomized. It was 32 patients, so smaller study. And so one of the beauties of an adaptable design like S1609 DART is within the exact same study, we went from 33 to 34 and then eventually 53 cohorts. And so we created a dedicated high-grade neuroendocrine cohort. We addressed some of the criticisms around testing for microsatellite stability, right? Because that was only available in a subset of patients. And these were 19 patients who had high-grade neuroendocrine neoplasms. All patients were MSS. And the objective response rate was 26% in this selected group, looking prospectively only at high-grade microsatellite stable neoplasms. The 6-month PFS rate is 32%. With the duration of response about 10 months and ongoing in patients with partial response. Again, the safety signal showed that we had no grade 5 toxicities here. Rash and dermatopathy were the most common, with a couple incidences of low-grade immune hepatitis. And we see here, similarly, that patients, regardless of the organ of origin, could have responses. And that what one would expect from immune checkpoint blockade, those patients that have response often have durable responses lasting months, if not years, because the actual drug is their own immune system. And so to summarize this data, S1609 is the first NCI-funded rare tumor immunotherapy basket study, studied, I guess, 53 rare tumor types with ipilimumab and nivolumab. The neuroendocrine cohort showed promising signs of benefit, and you saw some of the translational data earlier, where really the benefit was isolated to a subgroup of patients who had high-grade carcinoma versus basically no response in lower-grade disease. And then we were able to move that forward within the same study into a prospective, dedicated high-grade neuroendocrine carcinoma, adapting to some of the concerns while we were in study, which really is the power of the cooperative group system. Some of the other Cohorts of interest we heard about that I think the data are really telling are the angiosarcoma cohort. This really dichotomized into responses in patients with cutaneous angiosarcoma, which likely relates to UV-mediated molecular signature in terms of the types of neoantigens that are created, especially on the face and scalp. Meanwhile, the radiation associated had a lower response rate, but you see here at least one patient with breast-induced radiation sarcoma had a response. And so I think the translational analyses here are going to be really powerful. Because it turns out where the cancer happened isn't the full diagnosis. It's these molecular and immunomic features that are so key. Metaplastic breast cancer, so metastatic breast cancer, but metaplastic really is the teratoma in terms of having multi-lineage components to the breast tumor. This is from our colleagues, Sylvia Adams. And so she had a case report, a patient responded. About half these patients have PD-L1 amplification. So it's a genomic amplification, not the immunohistochemistry that you necessarily see for use in non-small cell lung cancer, other tumor types. And they typically are microsatellite stable, have low tumor mutational burden. And these patients can have dramatic responses. So this is a patient who basically their chest wall lesions, both by PET as well as on skin, had effectively a near complete response on treatment. And so looking at these patients, we see 3 patients with durable response, both in waterfall plot as well as Swimmer's plot here. And it didn't, the type of histologic predominant subtype, meaning whether a spindle predominant versus squamous versus other, didn't drive that analysis. I think yet again, histology is only part of the diagnosis. I think the genomics here and the translational work here are going to be really key in trying to discern which patients respond or not. A couple other cohorts of interest, these have been presented recently at ACR by my colleague, Dr. Young Chai, was clear cell ovarian cancer. It's a subgroup of ovarian cancer, actually quite difficult to treat, tends to be platinum resistant, though with the shortage of platinum, everything's being platinum insensitive right now. But the idea that there's a subgroup of patients who had benefit with dual immune checkpoint blockade only, no platinum-based therapy as part of this combination. And we see here durable responses in this subset of patients. This actually is another patient that I have the pleasure of seeing in my own clinic, and they're continuing to do well. And so really trying to understand the why behind it is I think where the study really is focused. And you heard from my colleagues some of the data. Another area which really is this is actually a New York Times article first, was about a rare type of cancer, small cell carcinoma of the ovary, hypercalcemic type, and a group of 30-something women who had this cancer actually really advocated get off-label use for PD-1 therapy. And they actually had a reasonably high response rate. It was obviously a small cohort, it's a rare cancer of 3, but 2 of them had near CRs. And so that led us to include this in the DART study because it's the quickest way of investigating this prospectively. And you see here again, there's a subgroup of patients that have really nice responses, including durable responses. This patient has the one you see here with on the Swimmer's plot has a 30-month PFS, right? So this is a patient otherwise who wouldn't have very many options above and beyond methotrexate-based regimens, which are traditionally used. And so understanding the genomic characteristics that belie this response, I think is a really important area. And then ovarian granulosa cell tumor, which is another subtype of ovarian cancer, quite rare. So now we're getting into the rare subtypes of more common cancers. We see here, again, a subgroup of patients, median progression-free survival almost approaching almost 5 years in one patient, right? And so basically, these are patients who've been on the study as long as we've been doing the study, and they continue to do well. And so understanding why I think is important, because these are both financially as well as medically toxic therapies. And so I think the biomarker story here is as important as the clinical one. I won't belabor the point on the collaboration with CMAX and immune profiling that's underway, but I think the clinical data really show that having a biomarker that can help you in clinic identify those patients that'll benefit would really be key. This is something that's been ongoing, and you heard this data from Kara and Young earlier, so I won't belabor the point, but the idea that this really is a national collaboration, right? So these are patients who really have participated throughout the country, including in neuroendocrine, where 2/3 of the patients were enrolled at community sites, and who basically, through the protocol, had specimens available, and through the CMAX mechanism, which is really national, had the data analyzed, and we're looking at this, try to identify those patients that can respond from our treatments. And we see here we're going to try to create the TCGA over tumors as much as possible, because even if there's not a clinical correlate that stands out, many of these tumors don't have sequencing data available in TCGA, and there may be recurrent molecular features that can help drive the next generation of SWOG and NCTN and investigator-initiated studies. And so this is something we're really keen to make public, because this data doesn't exist. There's no way to otherwise do these studies, as well as some of the data that you heard about earlier. we talked about multiplex immunofluorescence, and I'd be remiss to mention it any more than my colleagues. And so really an efficacy signal across multiple tumor types, high-grade neuroendocrine cancer, for which it's in NCCN guidelines now as an option for your patients, angiosarcoma, metaplastic breast cancer, and these select rare ovarian cancer subtypes to date. As I mentioned, there's these remaining open cohorts, though actually as of the time I'm giving this talk now, the study is closed to accrual. And I think the most important point is that clinical trials in rare tumors are feasible. The study was open at 800 sites throughout the country. 2/3 of the patients were accrued in a community setting. And we enrolled 798 patients across 53 cohorts in about a 3-year span if you consider chest time, right? There are some stops and starts related to COVID-19. The neuroendocrine cohort went from 32 patients enrolled in 3 months with that signal in high grade, basically allowing us to deploy as an arm by amendment a dedicated high-grade neuroendocrine cohort that enrolled 21 patients in 2 months. And so really the need is out there for patients with rare cancer. Want to thank my colleagues and also those in SWOG and CTEP for helping us put this together. It takes a village for a study of this magnitude. I have more emails to Dr. Megan Othis than I do my family members, according to Microsoft Outlook. And so apologies, but thank you. And thank you to everyone who's been involved, and especially to our translational colleagues, where I think we're going to start hearing the second chapter of this interesting novel, talk about really the biomarkers that belie the responses and resistance. And so with that, happy to take any questions if I have time.
I'm going to have to cut you because we're running late, but that was a fantastic talk. And I think one of the really gratifying things about DART was meeting this unmet need for rare cancers, and also what you showed, these subsets of patients that would never have had access, that have these years, they probably cured, long responses. So thank you for leading this, what I think was a phenomenal study. And I think it was open at over 1,000 sites, just to correct you. Yeah. But our next speaker is Dr. Aluwalia, who is a leader at Miami Cancer Institute and has instituted our CNS working group. The Early Therapeutics and Rare Cancers Committees has 2 children. Think of them as children, the sarcoma working group and the CNS working group. So thank you.
Thank you so much for the kind introduction. And I'm just trying to find my slides too. Okay, great. So again, thank you, Dr. Kazrak, for the kind invitation to house our working group in your committee, also this opportunity. And I just hope we can follow the footsteps of, Dr. Patel, you and Yang Chai, on what you've done in DART, which is, offering hope. And that's what we want our group to do as well. And I should also congratulate Dr. Davis. I really enjoyed her presentation. And maybe in the CNS working group, step behind her in the sarcoma working group. But we have a big meeting today evening. I would invite all of you to be there if you can. It's going to be in one of the Pacific rooms at 5:15. We've got a nice grant from the Hope Foundation to launch our first working group. We're getting experts from other cooperative groups to help us share their journeys on how we can work more diligently to create success stories like that. And I hope in 2-3 years, we can showcase some of the work that you're doing. So congratulations on that. And with that, I'm going to launch on some of the work that the CNS working group has been doing in working with the other cooperative groups. So the first study was actually here led by Dr. Chad Rustovan with Paul Brown. This was looking at avenues to help patients who've got brain mets in terms of radiation post-resection. So there's data out there that if you resect a patient with brain mets and do not follow that with any form of radiation, there's a fairly high recurrence rates. And typically these patients have been treated with stereotactic radiosurgery with one form of, radiosurgery. And now there's also data that fractionated radiosurgery, that means radiation giving over a course of a couple of days, is actually better than one shot. So this trial, after patients get resected, will get randomized to a form of radiosurgery, stereotactic radiosurgery, which is just one shot of radiation based on the RTOG doses, which can vary from actually 15 grays to 20 grays actually in this trial, or give fractionated radiosurgery. That means you can give the same radiation, but break it down. So it can be 27 grays in 3 fractions or 30 grays in 5 fractions. And actually, SWOG did quite well, and the trials enrolled well. Next, moving on to other aspects of radiosurgery, again, building on the brain mets aspect. This is the NRG-BN009 study. It's the phase 3 trial of salvage SRS or salvage SRS plus hippocampal avoidant whole brain radiation for patients with first or second distant brain relapse. Essentially, what we've found out is that whole brain radiation is associated with a lot of neurocognitive decline that has actually led to a lot of use of either radiosurgery or hippocampal avoidant whole brain radiation. What we find out in some brain mets that despite using radiosurgery, they're bad players. So these patients tend to have a faster recurrence. So the question is, if we have patients who we can identify are more likely to progress quickly after radiosurgery, can we give them hippocampal avoidant whole brain radiation along with SRS? And the concept that was developed to look at that was by Michael Chang from Wake Forest. And this is a trial that is led out of NRG, where SWOG is actually partnering up. And Dr. Chad Rusto, once again, our champion here. And this is built around this brain mets velocity. And what it means is the patients who are likely to undergo fastest recurrence will then be randomized to stereotactic radiosurgery plus hippocampal avoidant whole brain radiation, along with memantine. So memantine is a drug that can help you mitigate some of the neurocognitive decline that we see with radiation. And then these will be randomized to SRS alone. Moving along, this was obviously, we tried a few years back to get the DART cohort for glioblastoma. So I know Dr. Kurzrock, Dr. Youngcha, and Dr. Patel was really influential. And just like our sarcoma group today had a concept that Alliance actually beat them to it. Similarly here, we actually had a couple of trial concepts where NRG got one and then Alliance got the other one. But this is the, what I can say is the DART version of glioblastomas or TMB high gliomas. So the first concept is actually through NRG-BN007. This is a phase 2/3 study of ipilimumab and nivolumab, and you've heard enough about these drugs for me not to tell you more. But this is basically now looking at glioblastoma. So as you know, glioblastoma is the most common brain tumor, malignant brain tumor we see in adults. Survival in unmethylated glioblastoma is only around 13 months. And the drug that we use typically is the chemotherapy called temozolomide, which is associated with a 21-day benefit. So we give patients 9 months of chemotherapy for a 21-day benefit, okay? And it's approved. So this tells you how desperate we are for new treatments. So here, this is the DART version of ipilimumab and nivolumab, now combining it with radiation and comparing this to temozolomide. So the phase 2 component actually has been enrolled, and the trial is going through some interim analysis to see if the combo has efficacy, and if it does, it'll launch into a phase 3 component for FDA approval. Next on is this Alliance concept. Now, again, a DART story, but now this was in somatically hypermutated recurrent glioblastoma, and this was one we had really gotten a lot of headway initially with CTEP, and then at the last moment when we thought we would get it, we didn't, because Gavin Dunn and Eva Galanis through the Mayo Consortium's Alliance A071702, a mouthful, is a phase 2 trial of checkpoint blockade immunotherapy in patients with recurrent glioblastoma. And basically what it uses is the foundation medicine testing, and they define the tumor mutation burden as more than 20 mutations per megabase pair. And essentially, again, looking at combination of nivolumab, ipilimumab, just like DART. And if they have more than one response in the first 18 patients, then launches into stage 2 design. And I know Dr. Patel talked about that. So this could have been one DART study that we would have had here, but we are supporting this for the Alliance group. So 2 concepts that are going through our working group right now. Both of them are going to be looking at glioblastoma, and essentially for people who treat patients with glioblastoma, there were 3 large randomized phase 3 trials looking at nivolumab in glioblastoma, both in 2 trials in upfront glioblastoma and 1 trial in recurrent glioblastoma, and all those trials have been really dead negative till date. So the question is, can we do better with 2 IO combinations compared to 1, because anti-PD-1 is not that great of a target. So Dr. Tony Wang out of Columbia has been working on developing this concept, which is a phase 2 study of etolizumab along with tocilizumab with radiation in patients with newly diagnosed MGMT unmethylated glioblastoma. And essentially there's evidence out there that anti-IL-6 monoclonal antibody when used in genetically engineered orthotropic GBM murine models can be an exciting therapy, and it basically leads to reduction in the CD68 plus myeloid cell infiltration, which are immunosuppressive, but increases the CD8 T cell infiltration. And when looking at now mouse models, when you combine the anti-IL-6 along with anti-PD-1, here you can clearly see in the green curve a lot of synergy there, which leads to better outcomes. So utilizing that, we have a concept where we are hoping to enroll patients with unmethylated MGMT at diagnosis, again skipping temozolomide, which is the drug that is a 21-day benefit drug, so practically not much benefit. So we will skip that and we'll add radiation atezolizumab with tocilizumab followed by atezo-toci until progression. And essentially we are powering this to be a single phase 2 study looking at historical cohorts, and hoping to get it through CTEP rather than going through the malignancy steering committees, which all of us know is a really tall hurdle. There is some interest in this because CTEP actually approved a recurrent GBM study in NRG. So there is at least some emphasis that they like the combo. So this is our opportunity, and we'll discuss this further in our CNS working group today and discuss this with the statisticians, and then come back to Dr. Kurzrock and the committee to see how we can push this forward as a concept. And then the remaining patients, so when we look at patients with glioblastoma, 60% of them are unmethylated, but 40% of them are methylated. So we are creating a sister concept, and this is more genomically driven. So this is looking at TRC102 and temozolomide in patients with newly diagnosed glioblastoma. And here we are taking advantage of a genomic signature that we've actually developed, and I'll showcase that to you in the next few slides, through CTEP supported trial through the Adult Brain Tumor Consortium that we had for over 2 decades, and just got actually the funding was pulled back. But we ran, I was the PI of the study, we ran out of that consortium. And essentially this was built on that in MGMT methylated patients, actually temozolomide results in 6-month benefit. In unmethylated patients, it's only a 21-day benefit. But in methylated patients, when we use temozolomide, you actually cause a 6-methyl guanine, 7-methyl guanine, and 3-methyl adenosine DNA adducts. The MGMT component is repaired by of the 6-methyl guanine is repaired by MGMT, but the 2 adducts, which are the N-methyl guanine and 3-methyl adenine, are not repaired by that, and that's repaired by base excision repair pathway. So in a sense, we are trying to do a 2-hit process here. And so TRC102 is actually an academically developed drug, was developed by Stan Gerson, who's a cancer center director at Case Western, and essentially is a base excision inhibitor that binds to the apurinic side created through the action of methyl MPG. And essentially, we showed it in orthotopic xenograft models that when you use the combo, you actually have best survival. So this led to a study out of a CTEP-supported protocol through the Adult Brain Tumor Consortium, where we looked at recurrent glioblastoma patients who had progressed after radiation and temozolomide, and we gave them a combination of TRC102 and temozolomide. And unfortunately, like all the trials in recurrent glioblastoma, we did not see any success. But what we did was we actually found 2 exceptional responders, and we defined them as 2 patients who had progression-free survival in excess of 18 months. And what we did was we just actually did a whole exome and transcriptome on them. And what we found out was that actually MGMT methylated patients were getting more durable benefit with the 2-hit hypothesis, which is, intuitively makes sense, because if you're getting hits through 2 pathways, one is being repaired through MGMT, and you block that with temozolomide. The second is the base excision repair pathway. You block that through base excision inhibitor, which is TRC102. So here in the MGMT patients, you actually saw, even in recurrent patients, median overall survival of 30 months. To give you a perspective, that overall survival of glioblastoma is around 16 months. So now we're becoming a little bit more precision oncology based, if I can use that term, in glioblastoma. And in unmethylated, we were doing quite crappy, 10 months, right? Which is fine, because that's what we get typically in this patient population. So there is some synergy in methylated MGMT patients. So and we took it further, we did whole transcriptome. And what we found out was that at least these 2 patients, actually, here you can see them, about both that I treated. And these were the patients, one of them got a progression-free survival of 18 months. The other one was progression-free survival benefit was like addition in north of 30 months. okay, just to a frame of reference, these patients with recurrent glioblastoma die within 9 months of having a first recurrence. So there's this signature which existed in around 10% of patients. And this was an effort, a collaboration done with Mike Behrens at TGen. And essentially, both these exceptional responder patients had this gene signature. So on the basis of that, we are proposing a phase 1/2 study out of SWOG, where we want to do the study only in the MGMT methylated patients, but when we'll prospectively validate this biomarker. And this concept, again, will be discussed at the working group today at 5:15. So those of you who want to come, it's in the Pacific H&I, and you're most welcome. With that, I would also love to thank Dr. Kazrak for the support that she has given it to our group. And I hope we present this, we can at least have 2 concepts in development. That's my email. If you want to join the CNS working group, you guys are most welcome. We certainly can use some of the success we've seen through DART here for our patients.
Well, thank you. Yeah, so we'd love to see more in GBM. Obviously a rare tumor, a big unmet need. Are there any questions? Okay.
Thank you.
Thank you so much. Our last presenter, we have a really tight day today, very full schedule, is Dr. Riva Basho, who is going to talk to us about combo match.
All right, I'll try to keep this quick and short, but thank you for having me today. So I am here to chat about combo match. Let me just find my slides. So combo match, as you all know, is the successor to the NCI match study, this time looking at combination therapies. It officially launched about 2 months ago with 3 initial studies. SWOG is leading one of the initial studies, S3, which we'll talk about in detail today, but there are 2 other NRG-led studies, which I'll also briefly just present today as well. So as of yesterday, there were 39 approved sites for combo match. At this time, the enrollment remains 0. So the SWOG concept is a phase 2 study of paclitaxel and ipatasertib in taxane refractory participants with AKT-altered advanced non-breast solid tumors. Here's the schema of the sub-protocol. It will enroll patients with advanced non-breast solid tumors harboring a mutation in AKT 1, 2, or 3 who have progressed on prior taxane therapy. Patients with mutations in KRAS, NRAS, HRAS, BRAF, or prior AKT inhibitor treatment will be excluded. All patients will receive a combination of paclitaxel and ipatasertib. And just some brief background, as we all know, the PI3 kinase pathway is a intracellular signaling pathway that results in growth, and the AKT is a central node of the pathway. Alterations in AKT are less common than other alterations in this pathway, but are seen at a rate of about 2 to 3% across tumor types. Ipatasertib is a potent ATP-competitive small molecule inhibitor of all 3 isoforms of AKT with weak suppression of other members of the protein kinase family. Some preclinical and early clinical background on this combination. So in the NCI match study, ipatasertib was evaluated in patients harboring AKT1 E17K mutation. You can see the majority of the patients enrolled in this protocol were breast, but other endometrial and GYN cancer patients were also prevalent. This was a heavily pretreated group of patients, with the majority of patients having received at least 3 prior lines of therapy for metastatic disease. And despite this, the study reported a partial response rate of 22%, stable disease 56%, and a 6-month PFS of 44%. Now, looking at the combination with taxane on the right, preclinically, you can see in a PIK3CA mutant ER-positive breast cancer PDX model that the combination of paclitaxel and ipatasertib resulted in reduced tumor volume compared to either agent alone. Here's an early phase 1b study looking at ipatasertib and chemotherapy. This is an arm shown here looking at docetaxel in combination with ipatasertib. And you can see there was tumor reduction across tumor types. And if you look at the bottom, there was tumor reduction seen in patients who were both taxane refractory as well as taxane naive, and reduction seen in patients who harbored mutations in the PI3 kinase pathway as well as those who were wild type. And I'll draw your attention to the patient on the right, the extreme right, the breast cancer patient who was taxane refractory, harbored a mutation in AKT1, and had a significant reduction in tumor. The reason that breast patients are excluded from this protocol is that this combination has been evaluated in the phase 3 setting in breast cancer in patients harboring any alterations in the PI3 kinase pathway in both triple negative and hormone receptor positive breast cancer. And you can see that the combination was actually ineffective, but it's unclear if this is due to heterogeneity of the patients that were enrolled, particularly in the triple negative cohort. So there's biomarker analysis that's ongoing. We don't know at this time how many of these patients were AKT mutants specifically and what the response rates were in those patients. We do have language built into the protocol that if that data comes out, that we will reevaluate whether we would continue with the protocol based on the results that we're seeing. So going back to EAY-191S3, the study objectives, primary objective is to look at the overall response rates, secondary objectives, progression-free survival, duration of response, overall survivals, and adverse events. And translational medicine objectives, part of these are part of the larger combo match study, and then also cohort-specific evaluations. We'll be looking at whole exome sequencing, RNA sequencing, and IHC for P10, both at baseline and on progression. We'll also be looking at plasma AKT allelic burden, as well as other findings using ctDNA. And then if tissue is available, fresh tissue, we will be looking at RPPA to look at predictors of response and resistance. Here's the eligibility. Patients with metastatic non-breast solid tumors, performance score of 0 to 2. Prior progression within 6 months of taxane-based therapy in either the curative or metastatic setting. No prior AKT inhibition, prior PI3 kinase mTOR inhibition is allowed. Neuropathy resolved to at least grade 2, stable CNS disease, no leptomeningeal disease, and then there's some diabetes limitations for patients who are on insulin or have a baseline fasting glucose greater than 150 because of the AKT inhibitor. All patients will receive paclitaxel days 1, 8, and 15 out of Q28-day cycle, and abatacertib dosed at 400 milligrams days 1 through 21. Here are the anticipated AEs based on the two agents. As we would expect with PI3 kinase inhibitors, there's rash, hyperglycemia, and diarrhea, for which we'll be watching. And then just the statistics, we put the null hypothesis at 20%, sort of on the high end of what we would expect with taxane alone, and then alternative hypothesis at 40%. And I won't go into the details, but we do have a futility analysis built in for efficacy. Here again is the TM collection. We will be looking at baseline mandatory biopsies for whole exome RNA sequencing and RPPA if tissue is available, optional biopsies on progression, FFPE archival tissue, and also whole blood at various time points. So just presenting quickly the other two concepts that are currently open. Here's EAY-191N2. This is a phase 2 trial of fulvestrant and binimetinib in patients with hormone receptor positive metastatic breast cancer with a frame shift or nonsense mutation or genomic deletion in NF1. So here is the study schema: hormone receptor positive metastatic breast cancer patients who have progressed on prior CDK4/6 inhibition and have an NF1 disrupting mutation. Enrolled in one of two cohorts, fulvestrant naive or for fulvestrant exposed. The fulvestrant naive patients will be randomized, looking at fulvestrant alone versus fulvestrant and binimetinib, and the fulvestrant exposed patients will receive the combination. Again, they have to have hormone receptor positive disease with a non-functional mutation in NF1. Here's some preclinical data. NF1 mediates signaling through ER as well as through the RAS pathway, and loss of NF1 increases signaling through both of these pathways, providing rationale for this combination. And on the right, you can see that specifically in NF1 mutant cell lines, when MEK inhibition is combined with fulvestrant, this results in reduced tumor size. Expected AEs with MEK inhibitors in general, ocular and cardiovascular side effects that we will be watching for through the protocol. No overlapping toxicity is expected with the agents. And then the last protocol, EAY-191N4, is a randomized trial of selumetinib, another MEK inhibitor, with olaparib or selumetinib alone in patients with recurrent or persistent RAS pathway mutant ovarian and endometrial cancers. Here's the schema. It's a little bit complicated, but essentially any patients with ovarian or endometrial cancer with RAS pathway alterations, prior MEK inhibitors are excluded, and patients who have progressed on PARP are excluded. Patients will be divided into ovarian or endometrial cohorts, and within ovarian divided into low-grade serous ovarian cancer or other ovarian tumors. Within endometrial divided into endometrioid versus other. And all of these strata, patients will be randomized to the combination of selumetinib and olaparib versus selumetinib alone. And here's some preclinical rationale for this combination, essentially in KRAS mutant cell lines. There was a decreased response to PARP inhibitors, but when combined with MEK inhibition, we saw a decreased cell line survival as well as tumor volume in vivo. Here is the patient eligibility for this protocol. So patients should have progressed on first-line therapy for recurrent or persistent disease. Ovarian patients should not be eligible for further platinum-based therapy. Endometrial cancer patients should have received prior IO agent and unlimited prior lines of therapy. AEs, again, sort of what we would expect with the agents, but special monitoring for ocular as well as cardiac toxicities based on the MEK inhibition. And that is all I have.
Well, thank you. So any questions? Oh, right away. Maybe questions.
I beat you, Rafi. And, Ravia, thank you for the presentation and for being the SWOG champion with our first entry into combo match. When you registered downstairs, you probably saw there's additional materials about combo match, nicely put together by the NCI and our colleagues. During the process and after Ravia got her project approved and underway, the NCI became very concerned about being able to handle. There was a lot of logistical delays. There was a delay in getting the pilot study going out of ECOG. And so a hold was put on new concepts across the NCPN. That's now been lifted or is essentially being lifted at this point. So we're looking forward to additional SWOG entries, concepts, combination regimens. And if you are interested, have ideas, don't think any idea is too simple or too unlikely to succeed. We'd love to hear from you and reach out to me or Chuck Blanky, and we'd like to work with you to get into wave 2 and follow on the success of Dr. Basu. Thank you.
Thank you, Gary. Thank you for that presentation. So my question is more on the translational aspect. So there is some data that, in fact, people from the NCI have looked at where ipatasertib, when you look at the downstream signaling for the AKT pathway, the phospho-S6, the EBP4, ipatasertib does not seem to downregulate phospho-S6, from a phosphoproteomic marker standpoint. Have you guys, or do you guys have any additional data to suggest that, because genotype may not be the same as phenotype here? So have you guys looked at that, thought of that?
We haven't. We have thought about it. I mean, clinically, IPAT and CAPI performed, obviously this is cross-trial comparisons, but at least in the NCI match, they had fairly comparable results. And so difficult to really comment on that any further. And we did, as I presented in NCI match, we did see results in the AKT altered population. So we're sort of using that as the basis to look at this combination, but it's certainly something that we will need to look at and a reason for why we want to pursue the RPPA as part of this.
Thank you.
Other questions? So obviously very exciting new pathway for combinations, and our committee would be happy to be involved as we figure out how we're going to be involved. And thank you, everybody, for being here and for your participation in all our studies. And thank you. Have a great rest of the meeting.